This will be the final lecture or the final video from this PowerPoint. Um, so we'll start by reviewing the sodium-potassium pump that we covered in the previous lecture builds up a steep sodium gradient with a great deal of stored potential energy in it. Secondary active transport moves the substance against its own gradient, but the transport protein never hydrolyzes ATP directly. Instead, it's powered by a second substance, almost always sodium, that is moving down its own gradient through the same protein at the same time. That's why we call it indirectly powered by ATP. The ATP was spent earlier by the sodium-potassium pump, and the secondary transporter is essentially withdrawing from a bank account that someone else has already filled. Now, secondary transporters split into two categories based on relative direction of movement. A symporter, sim meaning together, moves both substances in the same direction. Now, the classic neurology example of this is the SGLT, or the sodium glucose linked transporter, which binds both sodium and glucose on the extracellular side, so on the outside, and using the energy released as sodium moves down its concentration gradient into the cell. It drags glucose with it. Now, even in situations where glucose is already more concentrated inside the cell than outside, a movement that would be thermodynamically impossible for glucose acting alone. An antiporter, anti meaning opposing or opposite, moves two substances in opposite directions, coupled within the same protein and the same conformational cycle. Now, the example most relevant to us is NCX, or the sodium-calcium exchanger. In this case, the sodium flows down its steep gradient, and that released energy pushes calcium out against its own gradient. Now, neurons depend heavily on exchangers like this to clear calcium that enters during electrical signaling. And we'll come back to this later once we reach synaptic transmission. Here's the point that I want to leave you with where this pump is concerned. Secondary active transporters never touch ATP, but their function is completely dependent on the sodium-potassium ATPase, continuing to run and maintain that sodium gradient. Now, if that gradient were ever allowed to collapse or dissipate, every sodium-coupled secondary transporter would immediately lose its entire energy source and stop functioning, even though none of them ever directly consumed ATP themselves. Now, primary and secondary active transport are not two unrelated categories that happen to share the word active. This is all one single interdependent energetic system. This is also why the cell is willing to spend so much metabolic energy running that one pump continuously. It is indirectly financing every single sodium-coupled transporter in the membrane. Now, a vesicle is a small, roughly spherical, membrane-enclosed sac that is built from the same phospholipid bilayer as the plasma membrane. It is used to transport material while keeping it physically separated from the surrounding cytoplasm or from the extracellular fluid. Because this requires the cell to physically bend or to pinch or to fuse sections of its own membrane, all of it requires ATP and the coordinated action of many proteins. Now, this process is inherently active, even though it doesn't fit neatly into a primary or secondary category, since it's not moving individual ions against a gradient through a single transport protein. Now, endocytosis is the general process of the cell taking material in. So a region of plasma membrane folds inward, forming a pocket that deepens. It pinches closed at the neck and then buds off entirely, producing a new independent vesicle into the cytoplasm. Now, the pinching off step is one continuous membrane splitting into two separate sealed compartments. That is called membrane fission. Fission, fission. Now, this requires a specialized protein called dynamin. which assembles into a ring around the neck of the forming vesicle and uses energy to mechanically constrict and sever it, much like a drawstring being pulled tight and cinched off. Now, we have different types of endocytosis. We can divide endocytosis into three basic categories. The first of which is phagocytosis, which phago meaning eating and cyte meaning cell, so it's cell eating, handles a large particle. So we're thinking a whole bacterium or cellular debris. The cell extends pseudopods. Pseudo means false and pod means feet. So it extends these false feet or these pseudopods that physically surround and engulf the particle before fusing and pinching off into a large vesicle called a phagocell. Some means body and phago means eat. So it's an eating body, basically. Now, in the nervous system specifically, this is carried out primarily by microglia. And we'll get into that more later. Another type of endocytosis that we have is penocytosis. Peno means to drink, and cyto means cell. So penocytosis, cell drinking, takes in small volumes of extracellular fluid non-specifically. So non-specifically meaning it doesn't really care what it's drinking. It's just going to drink whatever's there. The last type of endocytosis is receptor-mediated endocytosis. Now, this is the most selective. A specific receptor protein will bind tightly to a specific ligand. Now, what does that mean? The receptor itself is on the plasma membrane. A ligand is any particle that binds to that receptor. So once that ligand binds to the receptors will cluster together, and then a molecule called clathrin, C-L-A-T-H-R-I-N, clathrin, will assemble into a lattice-like coat that derives the membrane to curve inward into a clathrin-coated pit, which then pinches off into a clathrin-coated vesicle. This lets the cell take up specific targets with high efficiency, even at very low extracellular concentrations, which is a sharp contrast to penocytosis's indiscriminate sampling. Now let's talk exocytosis. Exocytosis is essentially the mirror image. It's the complete opposite of endocytosis. A vesicle that is already in the cytoplasm travels to the plasma membrane. When it makes contact, it will fuse with it, the vesicle's membrane merging completely into one continuous membrane with the plasma membrane. Now, this does not just happen because the two membranes touch. Bringing the two hydrophilic surfaces close enough to merge is energetically unfavorable. These specialized fusion proteins called snares, S-N-A-R-E-S, snares, sit on both the vesicle membrane and the plasma membrane. Now, they bind tightly to one another and physically pull the two membranes into extremely close proximity, destabilizing both bilayers just enough to let them merge. We will go much deeper into the snare mechanics in a later unit when we talk about synaptic transmission. Now, just like there are different types of endocytosis, there are also different types of exocytosis. There is constitutive exocytosis, which happens continuously and has no specific trigger. The vesicles form, they travel, and fuse as part of routine cellular activity. Then we have regulated exocytosis. Now, this withholds fusion until a specific signal arrives. Something has to tell the cell to do the regulated exocytosis. So vesicles are pre-made and they're docked near the plasma membrane. Fusion of these vesicles happens rapidly and all at once as soon as the trigger occurs. The single most important example of the entire nervous system is neurotransmitter release. It's specifically the arrival of an electrical signal causing calcium to enter the presynaptic terminal that triggers snare-mediated fusion and the sudden rapid release of neurotransmitter into the synaptic cleft. Now, that will be the central subject of an entire later unit. All I really want you to get right now is the constitutive and regulated exocytosis, and we'll talk more about neurotransmitter release later. Now, I want to end this unit by tying it back to where we started. Both endocytosis and exocytosis depend entirely on the same fundamental property that we established in Lesson 1, the amphipathic self-sealing nature of the phospholipid bilayer. Now, a piece of a membrane can only fold inward and pinch off, or a vesicle can only approach and fuse with another membrane, because phospholipids spontaneously arrange themselves to keep their hydrophobic tails shielded from water at every single step. Even these large-scale whole membrane movements, the kind that release neurotransmitters that let a macrophage engulf a pathogen, are ultimately governed by the same basic chemical logic that explained on day 1 why the bilayer exists at all.